Okay, so welcome back. Now it's a question answer session, and I would like at least from each group, like that means 2, 3, 4, and 5, you'll ask questions. So first option is for group 2 students. Please ask questions from the first group. Group 2 student, anyone from group 2?
Yes, ma'am. I had a question.
Yes.
Adish?
Yeah.
Ma'am, I had a question to Adish, but he's not here.
Adish, okay, others will try to answer. I guess it's a combined presentation. Go on. Manaswan is there.
Okay, so in one of Adish's slide, he has talked about viruses and virions. So what's the main difference between them?
Okay. Yes, Manaswani, can you tell, please?
Yes, ma'am, I can. So, first of all, like I had mentioned, see, a virion is what you used to describe when it's the entire segment together. So you have, let's say, the genetic material and then the protein coat, and let's say you have an envelope or a glycoprotein spike. So everything together, when it forms one particular unit and becomes infectious, then you call it a virion. So that this was also the first term that was given, and later only people started using the word virus, but the correct technical term for the entire unit is virion.
Okay, thank you,
Yes,
Manaswan.
that's right, Manaswani. Yeah, group 3. Who's asking from the group 3? Please come. Hurry up, hurry up. Group 3 people. You will get negative points for this if you don't answer, right? I told you earlier also that there are marks for different parts, for different portions in the entire presentation. So it will go as a negative. Who are in the group 3? You want me to open up the Amazon and check your roll numbers and call you by name?
Ma'am, I have a question.
Who's this?
Devanshi.
Yeah, Devanshi, go for.
Any other example, a common virus of plant and animal which infect humans?
Common virus which infect humans and plants both? You mean?
Plant and animal both infect humans.
There's no common virus that way. We learned it in class. A plant virus can't affect an animal.
Yes, Devanshi, there is no virus which can infect animal or a plant or yes, human and animals. Yes, they could be common viruses and pathogens, but there is no virus which can infect all three. I mean, plant viruses cannot infect humans. Viruses need certain receptors, right? In human and animal viruses, there are certain receptors are to be required in the host. So without those receptors, the infection cannot take place. Yes. Okay, fourth group, hurry up.
Excuse me, ma'am.
Yes.
Ma'am.
There was some news about the red pepper virus that it can
There was
infect.
some
News about the red
see.
pepper virus.
No. It was not infecting. I tell you, I've read that paper. That virus was found in the fecal extract of the human. So that means those that might have virus, and those particles were found in the fecal. That is, but replication is not possible. It is not infecting. Particles can be, it moved inside your body and then fecal matter is gone out, but it is not replicating there. It is not infecting, right?
Okay, ma'am. Okay.
Yes. Group 4 students, please go for this question.
Ma'am, I had a question for group 1.
Yes,
I
yes.
can't quite remember whose slide it was in, but someone talked about capsids and viral envelopes. So I just wanted to know any few differences they can list out between
I guess.
capsids and viral envelopes.
Yes.
Yeah,
Ma'am,
yes. Adish, can you answer? Or Manaswini? Anyone?
Yeah.
Fine if I answer, or Adish, do you want to go ahead?
Adish, you answer, please.
Okay, ma'am. Ma'am, okay, so capsid is basically the protein shell which protects the genetic material, and envelope encloses the capsid, and it's just another outer structure. Think of it like as the of the plant. And envelopes are made from proteins and phospholipids. And yeah, they sometimes they help in the fusion of host cell, as the envelope fuses with the membrane of the host cell. So they help in the infection.
Yeah. Yes, yes, Adish, you're right. So basically, not all viruses are enveloped. There are few viruses which are enveloped and few viruses which don't have this envelope, right? Okay, fine. Then group 5.
Yes.
Ma'am, I have one question. So I want to
Yes.
ask that what is cow mastitis test?
What is?
Cow mastitis test.
Yes, students, please go.
Ma'am, can I
Yes.
try? Like, it was included in
Yes, Sam.
Adish's
Yes,
slide. So
animal
basically,
viruses, I guess. Yeah.
it's California mastitis test, and it's used in to check in the cow somatic cell count of the milk. And if there are disrupting cell membranes present in the milk sample along the DNA,
Mhm.
I think it forms when reacted with a test reagent, it forms a gel. I'm not sure what is in the test reagent, but I know that it forms a gel if there is disruptions in the cells.
Yeah. It is basically some kind of a disease which is found in the cattle, basically, right? Which leads to the production of a abnormal or some abnormal kind of a milk. So this condition is prevalent in the cattle, right? Okay, fine. Tanvi, very interesting question. So can we just
Ma'am,
quickly
I
move to the next? Yes,
Ma'am, I have
Rupal?
a question for group 1
Yeah, please.
from group 4.
Okay,
From group
you are in
4
group
for
four,
group 1.
Rupal. Okay, okay.
Group 4, yes, ma'am. So what is the use of spike protein 19?
I could not hear your question. What is the?
Yes. Can you repeat?
What is the use of spike protein from in COVID-19?
Fine. Who
Ma'am,
will
can I
answer?
answer? Because
Yes, go.
I have already explained in last year in presentation. So basically, COVID has spike proteins. They are spike glycoproteins, and they use this to uh, I'd say they connect to the angiotensin converting enzyme ACE2 receptors, and then they introduce their mRNA inside. The RNA inside the cell. So it's actually a mechanism to enter the host cells.
Okay, fine. Yes, yes, that's right. Okay, any more questions, students? No. So
No,
can we
ma'am.
move to the next group, please? Next group will be group 3 or 2. I'm not sure. I don't know.
Ma'am, group 3.
Group 3, please upload your presentation and go on.
Good afternoon, everyone. Myself Janvi Singh, and my group is presenting on origin of viruses. We are Devanshi, Nandini, Kshitij, Ayushi, and me. So we're gonna start now. Next slide, please. Okay. So we know the earth never shoots beneath their feet. We have never found remains in the rocks, and by some standards, they are not even alive. Uh, Viruses are ancient and as simplest as they consist of genetic information, which is DNA or RNA, wrapped in a capsule of protein. They are just bits of proteins and genetic information, but we can also say they are a proof of being a very smallest thing that can have an outsized impact. We might get a sniffle for a couple of days or worse with these viruses. But these are those genetic burglars uh, which has no fossil record of viruses in the conventional sense. They are just too small and fragile to be preserved in rocks. But there are fossils of viruses of sort preserved in the DNA of the host that they have infected. But this molecular fossil trail can help us understand where viruses came from and how they evolved with the rest of us. And it can even help us to tackle the biggest question of all. Next slide, please. This biggest question is, are viruses alive? To consider this question, we need to have a good understanding of what is meant by life. Although many biologists have generally agreed that all living organisms exhibit several key properties or can function, like they can grow, reproduce, maintain an internal homeostasis, can respond to stimuli, and carry out various metabolic processes. In addition, population of living organisms evolve or over time. Well, in their origins through convention. Uh, sorry. Many of them are very small, of course, on in order of tens of nanometers, while others are surprisingly big. But they all rely on infecting some sort of host to reproduce and survive. We think that viruses have been around us as long as life itself, because they can infect all form of life, like bacteria, archaea, and eukaryotes, and because they are so simple, some scientists think they evolved alongside or even before the earliest cells. But without any real fossil evidence, how can we know that the history of what is the history of the viruses?
Janvi, your mic is on mute.
Yeah, your mic is mute.
Sorry. So, to order to look for traces of these ancient viruses, we expect to study the genome of their host. It makes sense when you think about how viruses actually work. Next slide, please. So, how viruses actually work? Viruses have to infect the host cell to access the machinery that it used to replicate its DNA, and then hijack that machinery in order to reproduce. In simple, these host cells is forced to manufacture new viruses, which then leave and look for new hosts to infect. Exception is virus and host don't always part ways entirely. Sometimes the genome of virus can become integrated into DNA of host. And as long as it doesn't cause a mutation that damage the host cell, the bit of viral information may stay there indefinitely. So, in the way, the viral genome become a sort of molecular fossil, and those ancient bits of viral information can also shed light on how old viruses are. That's because ordinarily virus can change really quick. Virus mutates so fast that after only a few hundred years, not much of original genome may be left. However, if the DNA is integrated into its host, then it can only mutate as fast as the host does. So mutation will be preserved, though not perfectly for way longer than a virus that just floats around there on its own. Next slide, please.
You are again on mute, Janvi.
I think someone is muting me. Okay. So now, as we get to know that these scientists can yet help to figure out the age of virus fossils, and they do it the same way the study of the evolution of other genes by lining up comparable sequence from different organisms and comparing them. If a sequence viral DNA is found in two different animals, then they probably both get it from a common ancestor. That means the virus has to be at least as old as the ancestor. This example is a circovirus, which is a group of a virus that is known to cause stomach problem in dogs. But scientists thought that it was scientists studied a bit, and then they get that it has been around less than 50 years. But traces later found in genome of dogs and cats and even pandas. So the viruses must date back to before those mammals, which could last shared a common ancestor, which might be as much as 68 million years ago. Next slide, please. So now, what is the oldest evidence of virus? So from the study of 2011, we looked at the history of Brachovirus, which usually infect wasps, and it found evidence to suggest that to group these viruses it could be as old as insect themselves, which is 310 million years ago. But other research has brought the history of virus even closer to home. Next slide, please. So now, if we talk about mammals and virus, the study was conducted in 2009, which found in mammals called in which a gene found, and this gene was named CGIN1 to the early days of mammals, which was between 125 to 180 million years ago. This gene is thought to be came from a virus, as part of it resembles a type of RNA virus, which is retrovirus. So some retrovirus infected a sperm of or egg of one of our mammal ancestors, which is billion of years ago, and now this gene is derived from is in us. Well, scientists think it doesn't function anymore, but it is one of the example how virus has left their mark on our DNA. It's been estimated that 8% of human genome includes a sequence that originally came from virus. So palvirology has helped us the date of the evolution of virus, which has hundred of millions of years ago, and now some of the models were introduced to us to help us knowing how the origin of virus uh, has developed or evaluate. Thank you. Next is Nandini, and I'm also open for questions now.
Okay, Janvi. Can you just tell me what are molecular fossils?
So molecular fossil is include the genetic information which is present in the fossils. So if we talk about viruses, we haven't find any DNA till now of virus, as it seems to be very small. But it has appeared in our DNA, and I've also given the example of this retrovirus, which is now being present in us as a proper sequence.
Okay, fine, Janvi. Can we have next student?
Am I audible now?
Yes, you're audible. Please switch on your camera also.
I'm switched on.
Yes, please start.
So I will be talking about the virus first theory. Viruses are intriguing biological entities that are borderline between the inanimate and living matter. They have RNA or DNA-based genomes with single and double-stranded nucleic acids, but lack functional translation machinery responsible for protein synthesis, including ribosomes and their own metabolism. They often integrate into cellular genomes and massively enrich the genetic repository.
of numerous organisms, including animals, plants, and fungi. Viruses are believed to have played important roles in the evolution of cellular organisms. Unfortunately, and unlike cells, viruses leave no fossil records. Their evolutionary trajectories must therefore be deduced from external viral features, a proposition that is problematic in itself. Historically, the question about the origin of viruses and life itself remains, for the most part, a philosophical debate and largely dealt with theoretical arguments rather than molecular data, especially because viral genomic repertoires are limited and patchy, like Janvi said in her previous slide, as viruses themselves were integrated into other cells in their DNA. So the only source of information was from their genetic materials. Next slide, please. So the virus force hypothesis states that viruses predated cells and contributed to the rise of cellular life. It was proposed by Conan and Martin, who in 2005 postulated that viruses exist in a pre-cellular world as self-replicating units. There is a major amount of viruses there, where all the viral genomes encode for the genetic sequences that lack clear cellular homologs. Presence of such virus-specific sequences provides support to their unique origin. This argument can be supported by the following reasons. First of all, the viruses were self-replicating units that evolved over the course of history. They became more and more complex, evolving traits and features such as cell wall and cell organelle. Secondly, it is already deduced by the researchers that the first gene of replication was in fact not DNA but RNA. There's also a point that these non-complex RNA strand-like organisms were the precursors of organic life. RNA molecules, ribosomes, exhibit enzymatic properties. They can catalyze chemical reactions. But all in all, contrastingly, all known viruses need a cellular host to replicate, thus necessitating the existence of cells before virus survival. Thank you.
Okay, Nandini. So it was about the virus first hypothesis. So you said that by some of the workers, this hypothesis was not considered very good or not in a supporting form for the viruses. So which points you mentioned, Nandini? Which 2 points were considered by the people that this is not the right hypothesis, which support the fact?
Ma'am, I'm sorry, could you repeat the question? I couldn't hear.
I just mean to say that this hypothesis was not accepted by a many group of people, and they gave some points in that this hypothesis is actually not the right one. So can you just tell me those points that why this hypothesis is not the
Ma'am, first of all, hello?
Hello, yeah, I can hear you.
Ma'am, first of all, I'm sorry.
Yeah,
I'm sorry,
can we have the next student here, please? Next student.
Yes, so I will be discussing about the progressive hypothesis. So, as Siddhav mentioned, according to this hypothesis, viruses originated through a progressive process. It describes viruses as derived from cellular RNA or DNA fragments which escaped from cells, such as plasmids, which are pieces of naked DNA that can move between cells, and transposons, molecules of DNA that can replicate and move around to different positions within the genes of the cell. When such RNA or DNA fragments acquired protein coat, they became independent entities capable of infecting cells from which they had escaped previously. As ancient RNA genomes were modular and were randomly distributed from cells to cells, during unequal cell division, a vesicle could have formed engulfing a self-replicating RNA and a coat encoding RNA segment. The translation apparatus was not transferred to the uniform vesicle, and thus it emerged as a RNA virus. This hypothesis is also called as vagrancy or the escape hypothesis. This hypothesis is buttressed by the existence of retroviruses such as HIV, human immunodeficiency virus. The genomic composition of retroviruses is remarkably similar to that of retrotransposons. As scientists studied the genomes of organisms, they found jumping genes called transposons, which were discovered in maize by Barbara Clinton in 1950. These genes can transpose themselves from one portion of the gene, like tourists hopping from one country to the next. Their copy paste mechanism was similar to the process retroviruses use. Thus, scientists speculate that these mechanisms may have found a way to not only copy their DNA to another portion of the gene, but to escape the cell itself, uh, turning into a full fledged virus. So now, to conceptualize this hypothesis, the evolution of a retrovirus from retrotransposon is illustrated. So since retroviruses are viruses that use RNA instead of DNA as the code in their genes, so a problem arises to infect us. They need to code switch to adapt to our rules that prefer using DNA as genetic material. So these viruses have no choice, as the enzymes that most organisms have are adapted to replicating DNA, not RNA. To perform this feat, they turn to one key enzyme, reverse transcriptase. So reverse transcriptase has a peculiar ability to stitch a complementary DNA sequence from an RNA sequence. So when the virus enters a host cell, this enzyme converts that single-stranded RNA into double-stranded DNA, which then migrates to the nucleus of the host cell, and by the help of integrase enzyme, the newly formed viral DNA gets inserted into the host cell genome. Viral genes can then be transcribed and translated, and hence virus replication commences. From this process, one can draw similarity to the movement of retrotransposons, which make up around 42% of the human genome. Like retroviruses, the viral-like retrotransposons encode a and so this escape hypothesis for the emergence of RNA viruses as well as small DNA viruses was advocated by many scientists, including Nobel laureate Howard Martin Taman. And a good example of viruses being created through gene escape events also includes human hepatitis delta virus HTV, which shows a similar ribosome sequence to the ribosome present in a human intron. HTV is found in only in humans, and it requires human hepatitis B virus to replicate. So this was the end of my part.
Okay, Ayushi, you presented really well. My question to you is also same as I asked Nandini, that what were the drawbacks of this hypothesis?
Ma'am, uh, if you talk about the drawbacks of this hypothesis, it fails to prove how viruses evolved from plasmids and how they acquired the ability to encode capsid proteins. And it fails to explain the presence of structures that are unique to viruses.
Yes, it fails to explain the complex structures of the capsids and other in viruses, that how they are so complex then if we are talking about this hypothesis. Okay, fine, Ayushi. Yes, can we have next student?
So, am I audible?
Yeah, you're audible, Dimanshu.
So, in contrast to progressive hypothesis, there is another hypothesis which is the reduction hypothesis or regressive theory, or we can say degenerative theory, which states that viruses resulted from the reduction of unicellular modern organism which can be bacteria, archaea, eukarya, to parasitic forms, or in other words, we can say viruses involved from more complex possibly free living cellular organisms, which is independent life forms, over time through a loss of essential functional genes. This means that cells of unicellular organisms lost their membranes, cell walls, and other structures, as well as their cytoplasm, hence retaining only the nucleic acid and some vital proteins in order to live in other cells as parasites. Some microbiologists, for the most part, concur that specific microscopic organisms that are commit intracellular parasites similar to chlamydia and rickettsia species advance from free living precursors. Another example can be leprosy bacillus. Like for we take an example, genomic studies indicates that the mitochondria of eukaryotic cells and Rickettsia provazekii may share a common free living ancestor. Also, it follows that existing virus may have evolved from more complex possible free living organism that lost genetic information over time as they adopted a parasitic approach to replication. Next slide, please. Thank you. So to illustrate this hypothesis in better way, let's take an example of nucleocytoplasmic large DNA viruses, which is NCLDVs. These viruses include smallpox and other recently discovered, which is mimivirus. Let's talk about mimivirus. Mimivirus is a genus of a giant viruses in the family of Mimiviridae. Its natural host is amoeba and it contains a single identified species which is named as Acanthamoeba polyphaga mimivirus, which is APMV. A typical brick-shaped pox virus, if we compare mimivirus, a typical brick-shaped pox virus have diameter around 200 nanometer, whereas mimivirus has diameter of roughly 750 nanometer. Also, mimivirus has a genome of 1.2 million base pairs to compare. If we compare with the poliovirus, it has a genome of only 7,500 500 nucleotides in total. So in addition to their large size, the NCLDVs exhibit greater complexity than other viruses have and depends less on their host for relationship than do other viruses. Also, some virologists have hypothesized that these viruses may be descendants of the more complex ancestor, as we mentioned in the case of mitochondria and rickettsia. So, according to the proponents of this hypothesis, autonomous organisms initially developed a symbiotic relationship. Symbiotic relationship, as we learned, one provides shelter and other provide food. Over time, this relationship turned into parasitic as one organism became more and more dependent on the other. As the smaller organism became more and more dependent on the larger organism, it lost its protein building structures. Eventually, it was able to replicate independently, becoming an obligate intracellular parasite as a virus. Thank you.
Okay, Devanshi. So, what were the drawbacks of this hypothesis then?
Ma'am, drawback for this hypothesis that if we talk about the progressive process, it argues that the retroviruses, we can take example, mobile genetic elements gain the ability, but of in the case of regressive, that it still it explains that the cells existed before the virus. So it's again the questionable that from where virus came. And this also explains the parasitic form of relationship, not the single form which the virus came from.
Okay, yes, can we have the next?
Ma'am, so my topic is on correlation hypothesis and chimeric origin hypothesis. It is correlation hypothesis, also called as bubble theory. Uh, in correlation hypothesis, it's majorly it's on viruses that virus and cells which have different origins and which are mutually influence their evolution. At the beginning of life, a community of early replicons or which is existed is approximately to a food source such as hot spring or hypothermal vent. Uh, the food source also produce lipids like molecules self-assembling into vesicle that could enclose replicons. Uh, its evolutionary process pressure is also could push replicants along two parts of development, that is first merging with the vesicle, giving rise to the cells, and next other is entering the vesicle, using its resources and multiplying, uh, and after that leaving it from another vesicle, give rise to new virus, give rise to viruses. Um, and in chimeric origin hypothesis, it is based on the analysis of the evolution of the replication and structure of molecules of viruses, in which a chimeric scenario chimeric scenario is for the origin of the viruses which is proposed in 2019. According to this hypothesis, the replication modules of viruses is originated from the primordial genetic pool, although the long course of their subsequent evolution involved many displacement by replicative genes from their cellular host, and by contrast, the gene encoding major structure proteins evolved from functionally diverse host proteins, and they throughout the evolution of virusphere, in which the uh, virus is uh, intact. And next slide. In conclusion, it is basically that what are the three main hypotheses aim in to explain the origin of viruses. The first is virus. The first hypothesis state that the viruses predated cells and contributed to the rise of cellular life. Uh, and it is merely contrastingly, all known viruses need a cellular host to replicate. The thus necessitating that the existence of cells before survival, and it is mainly first hypothesis which was erased the definitions in the way they are required for cells. And in the next, which is progressive or escape hypothesis, it states that the virus arose from genetic elements and that gain the ability to move between other cells, and it also fails to explain the presence of structure which are unique uh, to viruses and are not present in cells. Furthermore, some available data try to contradict the escape hypothesis because of the lack of homology in the protein sequence of the virus with their host. Last is regressive or reduction hypothesis. It asserts that the main remnants of the cellular organism. However, many components of how this process also occur may remain a mystery because it is does not explain the keys why the even the smallest part of cellular parasite do not resemble. And the main, so that's why it is very unclear for the origin of viruses because it do not form fossils. Till date, there is no clear explanation of viruses, uh, which is whether they are exist or not. Perhaps or viruses are arose, and it is by the mechanism which is uncovered and basic research in the fields like microbiology, genomics, and structure biology is mainly provided to answer the uh, basic question. That is my thank you.
Okay, Kshitij. So my question for you is, can you just tell me what does replicons means? In the co-evolution hypothesis, you said that viruses might have originated from the replicons. So what are these replicons? No,
Ma'am,
Kshitij?
replicons is basically the protein of the DNA which is uh, replicate in the viruses because viruses replicate much faster than bacteria and other organisms. That's why the
And
replicons.
what is okay, my next question is, what is virosphere? Virosphere.
And can you repeat?
Virosphere. You use the term virosphere when you were compiling your answer, that theories. Okay.
Ma'am,
So can we?
it's
Yeah. Yeah, please.
mm, ma'am, it's basically the virus are basically virus. Am I don't know that.
It's
So
fine. It's fine if you don't know, Kshitij. You can say that. No problem. Yes, the question, this is open for questions to everyone. So, group first students, please ask your questions.
Ma'am, is it possible if I go later? Because from the first.
If is it possible? What?
If I
Yeah,
can
it is for anyone from group one can. Anyone
no,
can answer.
I am still.
Anyone can question from the group one. Yeah.
So actually, I had a doubt from the bubble theory. I think that's what was the name. So in that, it was said that like viruses were replicons, and replicons are nucleic acids, right? So won't that nucleic acid be denatured? Because it said that they were in hot vents, something like that, thermal vents. So if like obviously there are thermus aquaticus, so if they were heat resistant, then how did virus survive in regions where like below the 50 degrees Celsius?
Yes, who will answer this?
If you want me to frame it in a better way, I can.
No, I understood your point. Yes, from group, this was the group three presenting, right? So anyone from group three can answer this question. Yes, Devanshi, can you answer this?
Ma'am,
Ideally, the yeah.
if we talk about the humans, they usually have the normal body temperature. So they think the human they can easily survive the temperature.
Anyone else from group three?
Yes, ma'am.
Yeah.
Uh, Ma'am, I have a question for Devanshi. Ma'am, so what's your current opinion on the origin of viruses?
So recent studies came that I don't know about the other one, but the Bamford has recently suggested that life started with the proteoviruses, which infects non-living vesicles, which further became protocells. But also in this scenario, protoviruses may have been they suggested that they have been membrane surrounded RNA recently. This information came.
So
That's
this was in some paper, Devanshi, you read, or it is just somewhere
No, ma'am,
in
I was
Wikipedia?
actually reading and a paper, I don't know where, on the Google Scholar maybe. Uh, in the recent researches, this was mentioned. I don't know, two universities or two companies' name were mentioned, but I only know about the Bamford.
Okay, fine. Yes. Next group, please.
Ma'am, my question is for Devanshi that so Devanshi, could you state some of the evidences that supports your hypothesis that viruses arose from bacterium?
Yes, I mentioned about the rickettsia and chlamydia. Um, Both are bacteria. In the case of chlamydia, it cannot reproduce by its own. Or in the case of rickettsia, the replication cycle of the rickettsia is the similar to the retroviruses. They affect the healthy cell and it can replaces its genome with the host cell that move into the next cell. So these two.
Okay, so any other group left for the questioning? I guess
Yes,
group
ma'am, I have a
5.
question.
Yeah.
Ayushi, you discussed about the similarity of movements of transposons with the viruses in the progressive hypothesis. So, could you discuss how the same goes with plasmids?
Yeah, like so I discussed about the movement of retrotransposons similar to that of viruses. So if we talk about plasmids, uh, plasmids as viral precursors, they also explains mechanisms found in both plasmids and in viruses like rolling cycle replication. Actually, I didn't find much material in this part, but there was one research article that described about one of the archaeal plasmid, which was extracted from an archaeal species from Antarctica. So, and which show that transfer mechanism, which was similar to the virus, so that the plasmid it encodes proteins found in membrane vesicles which enclose the plasmid DNA, and those vesicles they were capable of infecting a plasmid free state, and this plasmid was uh, able to integrate and replicate as part of the host genome. So it could resolve out with the fragments of host DNA or the portions of the plasmid left behind, form vesicles and uh, transfer to new hosts. So this mechanism of DNA transfer could represent a strategy used by viruses and can support a strong uh, evolutionary connection between plasmid and viruses.
Okay, Ayushi, that was well explained. Just one more question for you. What is difference between retrotransposons and transposons? There are two terms, right? How these two terms differ? Any idea? Anyone else from the group?
It is a type of transposon.
Yes, type of transposons. So, I mean, how does it differ from the transposons? Why it is called a retrotransposons? There must be some reason that it is called retro. Okay, fine. So Sam, your answer is still not discussed. since it's the time for the class tomorrow, we'll discuss your answer.
Ma'am, I had a question.
I had a
Who's
question.
this? Oh, बहुत सारे question है.
Uh,
Come on, shoot up.
I'm from group 4, ma'am. I just had a question for, I mean, not in specific, anyone can answer. Can anyone give me an example of an oncogenic virus?
Yeah, maybe even I have the same question.
Oh, guys, come on. Yes, what are oncogenic viruses? Can anyone name? I guess introduction part से, yeah, other groups can also name this. There are many viruses which are oncogenic. Can you just name any one? Yes, Devanshi, go on. Yes.
Ma'am, it's human papilloma virus which cause cancer in cervical.
Yes, it is very. Yeah, and large number of women population is affected by the cervical cancer, and lot of research is still being going on this particular virus, that how it can be managed and what are the steps that can be taken to manage this virus. Yes, good. Yes, anyone? There was lot of.
Yes, ma'am. Devanshi, just now you have talked about
doesn't
protovirus.
Yes. One by one. Yes.
Devanshi, just now you have mentioned about proteovirus. So can you explain it?
Proteovirus are free-living viruses. They are ancestral virus. We can say hypothetical primitive. It I don't know completely. It has the hypothetical sequence of chromosomal DNA in a somatic cell that can replicate by reverse transcription and also give rise by mutation to, which is a oncogenic virus.
Yes, there was a question from Adhish or someone else who was asking about HIV. Who was that? Please go on.
No, ma'am. I was asking, doesn't HIV also causes cancer?
HIV directly does not cause cancer, but yes, people who are affected by HIV, they are at a more risk for the A hepatitis B and oncogenic viruses, which include hepatitis B and C, other viruses also. So it is directly not causing oncogenic effect, but yes, people who are affected by the HIV, they are more prone to oncogenic viruses, right?
There's also simian virus 40. It causes oncogenic DNA. It's an oncogenic DNA virus.
Yeah, there are a number. Oh, fine. Yes. Any other question? Please go on.
Yes, ma'am. So I want to ask that.
Tanvi, you are in which group?
I'm group 5.
Group 5. Okay, Tanvi.
I want to ask that why human hepatitis delta virus requires hepatitis B virus to replicate, and why it doesn't require any other type of hepatitis to replicate.
Yes, students. Can you answer about hepatitis delta virus?
Ma'am, can I answer?
Yeah, please.
So I didn't go much in detail, but what I know is that hepatitis delta virus HDV, it is a satellite virus of HBV, so it requires its envelope glycoproteins for its infection cycle and transmission.
Yes, that's right. This is the answer. Anyone else? Hepatitis delta viruses were earlier thought to be like a virus-like thing, because when not much information was available, so once they thought that they are the viroids which are affecting humans, but later then a lot of studies were done, and then it was named as a new virus, not as a viroid but as a virus. Yes, anyone else? Okay, fine. It was a good interactive session, and I'm really happy that you all did a good job and worked really hard, and presentations were good. And maybe tomorrow we'll have one presentation, maybe group 3, group 2, right? We missed group 2 today.
Yes, ma'am, group 2.
So tomorrow we'll have a group 2, and then next Thursday we'll have the rest of the group which are left, because Monday, Tuesday I want to go for the lectures. I don't want to miss your lecture sessions basically, right? So tomorrow we'll have group 2, so one group and some discussion we can do combinedly tomorrow. and then the rest of the group on the next Thursday in the practical class, right? So please, for your attendance, switch on your cameras so that I can quickly go through the attendance. We have Adhish Mishra,
Present, ma'am.
Ayushi Jain,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi Sharma,
Present, ma'am.
Ishita Arora,
Present, ma'am.
Janvi Singh,
Present, ma'am.
Kshitij Singh. Yes, Kshitij, can you switch, just unmute yourself,
Present, ma'am.
please? Yes, Manaswani,
Present, ma'am.
Manaswani, Namrita, yes, you're there.
Present,
Nandini
ma'am.
Raj, Nehal Joshi,
Present, ma'am.
Rupal Joshi, Sahiba,
Present, ma'am.
yes, and we have Shakti, Sanjana,
Present,
yes,
ma'am.
Sanjana, Shakti,
Present, ma'am.
Shivani, yes, you're there.
Present,
Tanvi,
ma'am.
Pradeep, Vivek,
Present, ma'am.
yes, then after Vivek, we have Yukta and Yuvraj.
Present, ma'am.
Present, ma'am.
Okay, fine, fine. So, yes, okay. So, is there any survey being floated for joining back the campus? Because I think there is some kind of rotation going for the offline classes now.
Ma'am, only the survey that we are interested to come